Welcome you all. Today we discuss the last part in your unit Palynology, that is fourth unit in paper second. So next point in this topic is use of palynology in coal exploration. In last lecture we have discussed about the role of palynology in oil exploration. So likewise similar these palynology that will be also used in the coal exploration. As you know, coal, which is a very important mineral, and it is used or it is having very great economical value as it utilized for the energy generation, then productions of the steel. Also, it will be used in the chemical production. So, this coal that will be very economically important mineral, this coal that will be produced by the organic remains of the plant when these organic or the dead material of the plant that will be anaerobically decomposed at that time these black compact stony mass that will be produced, and that is nothing but the coal. So anaerobically when these organic plant material they will be decomposed and they are formed the black compact stony, and it's nothing but the coal. So when the explore of this coal will be takes place in the field, there will be explorations of the coal that will be very much will be depending on the palynological knowledge. So how this palynology that will be helpful for the explorations of the coal. So mostly the coal formation that will be takes place in the Carboniferous period. So according to the plants of the Carboniferous, these coal seams that will be identified, and the explorations of the coals will be takes place. So next point that will be methods of the coal explorations. So when the pollen grains or spores that all are present within the when the plant material that will be decomposed in the uh, that will be decomposed at that time the pollen grains or spores they will also will be dead, and they will be converted into the sediments. And when we deposit this coal, at that time the different varieties of these spores, different shapes, sizes, organization or ornamentations will be observed in the coal. So characters of these different characters of the spores or pollen grains uh, that will be found at the time of Carboniferous period that will be observed in the coal. So such a great taxonomical significance, and thousands of the different types of spores or pollen grains that are present in the microfossils. So when we explore the coal at that time, we can find out the different types of pollen and spores in the microfossils. So the quantitative or qualitative representations of such structures and their variations uh, that will be mostly observed in the different layers of the coals. So the part of the region under this study is marketed by trading samples. So how these pollen or spore analysis will be takes place. So for it, the concentrated HNO3 that will be adding on the concentrated nitric acid that will be first of all they will be added on these coals uh, of the with this nitric acid. The NaOH are also adding that coal. So alkali is to fed by constant washing with the water and the spores that will be mounted in the glycerin jelly. So as you know, this coal and that is the one type of rock. So First of all, when we undertake the study of the pollen and spores on the microfossil, first of all उस रॉक को हमें ट्रीटमेंट देने की रहती है, तो उसको क्या कहते हैं, नाइट्रिक एसिड और NaOH से उसे ऐड किया जाता है उस रॉक पे, फिर वाटर से उसे वॉश किया जाता है, एंड देन द पोलन और स्पोर्स which those which are present in these rocks, they will be separate out. Then these pollen and spores you can mount on the slides with the help of glycerin. So this is the figure which shows different types of spores which are present on the rocks. So same like the oil, mean microfossils index of the some coal example. So according to the types of coal, the different types of the spores will be present. So in case of the first grade coal, and there will be mostly Lycospora spores will be present. Then in second grade coal, there will be Calamospora spores are there. Then in third grade, there will be punctatus spores are present. So this is the identified spores which are present in the Carboniferous, or these are the Carboniferous plant spores. So if we found this type of spores in the rocks, we can easily confirm that this is the coal explored site. And according to this study, the exploration that will be started out. So in this way, this palynology that will be helpful in the coal exploration. Then next question is regarding the significance of the palynology in allergy. So as you know, in air, different types of pollen or spores that will be present. And these palynological study, in palynology we have studied about, or we analyze the pollen and spores which are present in the everywhere. So in aeropalynology, that means it is the term or it is the branch in which it is the science branch, and in which there will be study of the pollen and spores which are present in the air. So let's discuss about the significance of the aerobiology or aeropalynology in allergy. So allergy, as you know, this allergy is nothing but the conditions of the hypersensitivity of the body. Some humans body suffers from itching, then irritations, redness, rashes, patches like reactions due to the certain pollens or spores. That is the allergy. In certain cases, this allergy may be affected on the respiratory tract. Then the respiratory tract become the red in color. Then they will be swells out. Then there will be no do any functions. Sometimes the exhalations due to certain spores or pollen grains will be takes place. So in this way, the study of the aeropalynology, it is very important to find out such a pollens or spores that will be affected to the human body. So what are the sources of the allergic pollen grains? So mostly the pollen grains of the amaranthus, then cassia, prosopisia, parthenium, xanthium, holoptera, then cynaron. These are some common examples. In that, the pollen grains of that plant, they will be affected or they produces the allergies. So mostly the pollen grains of the Poaceae family, or which is also called as Gramineae family. That means the all types of grasses. So these Poaceae members, they will be dominant in all the regions of the India. In that, pollens of the Poaceae is about 20 to 45% in total aerospora is highly allergic. So in total aerospora, in that these Poaceae family plants, they are having the 20 to 45% of the spora. एयर में यदि हम जो भी पोलन और स्पोर स्टडी करेंगे तो उसमें से 20 to 45% जो भी स्पोर्स है वो हमें ग्रामिनी फैमिली के मिलेंगे और these pollen that will be allergic. Instead of that, some anemophilus, that means the airborne pollens of the Azadirachta, then plant Acalypha, Lantana camera, Argemone, Bougainvillea, Mangifera, then Brassica, etc. plants, pollens, they are highly allergic. And these pollens of that plants have proved that they will be allergic even through occurring in the low concentration in the air. यदि ये जो प्लांट्स है उसके जो पोलन्स है वो यदि लो क्वांटिटी में भी प्रेजेंट है तो वो एलर्जी प्रोड्यूस करते हैं. Instead of that, some peoples may have allergy with the dust. As you know, dust की allergy तो हर एक को रहते हैं. यदि हम dust में वर्क करते हैं तो हमें जो भी inhalations वगैरह सर्दी वगैरह हो जाती है. तो this dust, because of why? Because the dust contains some fungal pollen grains like the penicillium, aspergillus और alternaria. जो हम डस्ट रहते हैं डस्ट में कुछ फंगल स्पोर्स प्रेजेंट रहते हैं. ड्यू टू दैट फंगल स्पोर्स वी हैव affected or we have gone through the allergies. So in this way, these are the different source in the air due to the allergies or they will be affected to the human body. Then next is about the methods. So how these pollens or spores which are present in the air, they will be studied out. So for this study the allergies pollen or yellow allergies, the first method is used by the vacuum pump method. So in vacuum pump, just the air is drawn through this filter paper by using vacuum pump filter paper and they are changed after the 24 hours. So here in beaker just at the upper side adjust the filter paper and at the bottom of this beaker adjust the vacuum pump. So when we started this vacuum pump, the air will be gone through this filter paper and all the pollen or spores which are present in the air automatically they will comes in the beaker and then they will be studied out. Then second method for the pollen analysis which are present in the air, that is gravity slides method. So this is very simple method in that just use the gravity slides. It is like the simple slide but they are having some gravity or some dish like some small openings or dish like structures are there on these slides. So on these slides just spread out the glycerin jelly at the both sides and this slide will kept out in the air. So automatically when we जो एरिया में हमें पोलन एनालिसिस करना है उस एरिया में वो स्लाइड रखेंगे तब वहां पे जो भी पोलन्स और स्पोर्स रहेंगे वो उस स्लाइड्स को अटैच हो जाएंगे क्योंकि हम उसके ऊपर ग्लिसरीन लगा रहे हैं. तो ग्लिसरीन को जो भी पोलन और एयर पोलन और जो स्पोर्स एयर में प्रेजेंट रहते हैं वो उस स्लाइड को अटैच हो जाएंगे चिपक जाएंगे. तब फिर हमें 24 आवर्स वो हमें स्लाइड एज इट इज वैसे ही उस जिस जगह का हमें पैलिनोलॉजिकल स्टडी करना है वहां पे हम वो स्लाइड्स रखेंगे 24 आवर्स के. फिर वो स्लाइड्स हम निकालेंगे उसे हम माइक्रोस्कोप के अंदर देखेंगे तो वहां पे जो भी पोलन ग्रेन्स और स्पोर्स हमें डिफरेंस वहां पे नजर आएंगे. तो उससे हम आइडेंटिफाई कर सकते हैं कि इस एयर में कौन कौन से प्लांट्स के स्पोर्स है या फंगल स्पोर्स है वो सब हम विद द हेल्प ऑफ ग्रेविटी स्लाइड्स मेथड हम स्टडी कर सकते हैं. देन so these are the methods to study the different aeroallergens which are present in the air. So that will be helpful to correlate the sporadic aeroallergy, the frequency of aeroallergens. Some varies with the seasons according to the data. Aeroallergies, the doctors can be treated the पेशेंट तो उस यदि हम उस एरिया का एरोपेलिनोलॉजिकल स्टडी करेंगे तो उस एयर में कौन कौन से स्पोर्स है वो हमें पता चलेगा और उससे जो डॉक्टर्स है डॉक्टर्स को भी हेल्प मिलती है कि वो इजीली फाइंड आउट कर सकते हैं कि किस चीज की वहां पे पेशेंट को एलर्जी है. Sometimes the pollen grains that will be discharged the kinds of the proteins in the human body and under the favorable conditions. So in sensitive individuals, they can create the hypersensitivity and allergies. Proteins have different physicochemical structure, different molecular structure, and every sensitive individual response of these allergic substances differently. So when these spores or pollen that will be कांटेक्ट विद द ह्यूमन बॉडी तब क्या होता है वो पोलन क्या करते हैं प्रोटीन या सब्सटेंस वहां पे प्रोड्यूस करता है एंड ड्यू टू दिस प्रोटीनियस सब्सटेंस जो ह्यूमन बॉडी है ह्यूमन बॉडी में वहां पे सेंसिटिविटी क्रिएट होती है सेंसिटिविटी इन द सेंस हाइपर सेंसिटिविटी वहां पे क्रिएट हो जाती है और उसकी वजह से हमें वो एलर्जी हो जाती है उस चीज की तो जो एलर्जिक सब्सटेंस है allergic substance के जो structures as well as the molecular size that will be different and this will be जो प्रोटीन जो पोलन ग्रेन से जो प्रोटीन ऐसे सब्सटेंस प्रोड्यूस करते हैं तो हर एक बॉडी को सेम अफेक्ट नहीं करते तो जैसे हर एक इंडिविजुअल की जो इम्यूनिटी है उसके हिसाब से वो उस बॉडी के ऊपर रिस्पॉन्स करते सो and these allergic components that include or they produces fever asthma bronchitis some skin diseases also some seasonal allergies are also produces like but mostly in this pollen grains mostly asthma they will be produces 6% and rhinitis about the 40%. So when in the seasonal plants or seasonal allergies like in रेनी सीजन्स यदि हम कुछ प्लांट्स के कुछ कांटेक्ट में आते हैं तो हमें उस प्लांट्स की एलर्जी हो जाती है तो मोस्टली उसमें जो यहाँ पे दिया है अस्थमा और राइनिटिस वो मोस्टली वहाँ पे एलर्जिक है और बहुत सारे जो पेशेंट है उससे अफेक्ट हो जाते हैं सो दीज seasonal plant they will be mostly produces the allergies so aeropalynology study that will be helpful or useful to collect the data of the various aeroallergens in its seasonal variations then they also affected on the aeroallergens on the humans and also the other organisms and remedies for these allergens obtained by the aeropalynological. So due to these different allergens, mostly the seasonal plants, they will be mostly created these allergens substances and due to these substances the allergies that will be produced not only in the human body but also the other organisms they are also caused or also produces the allergies so in this way these study of the aeropalynology it is very important with the help of aeropalynological study, we can easily find out the pollen grains and according to this pollen or allergy, doctor can suggest the or doctor can give the treatment. So in this way, the palynological study or aeropalynological study that will be very important. Then last one, it is the role of palynology in forensic science. So, as you know, little bit idea about the forensic science that will in solving the different types of the cases or legal cases, these study of the pollen and spores that will be helpful. So studies of the polymorpho trapped in material associated with the criminal or civil investigation for investigating the criminal sites or the used material or at the time of crime. These palynological study that will be takes place. जो crime होते हैं different types के सुसाइड है या किसी का खून हुआ है तो एट द टाइम वहाँ पे जो भी मटेरियल यूज किया जाता है उसके ऊपर पोलन्स या स्पोर्स उस एरिया के अटैच हुए होते हैं तो जो पैलिनोलॉजिकल स्टडी है वो मटेरियल का पैलिनोलॉजिकल स्टडी किया जाता है और वो जो इन्वेस्टिगेशन है वो हमें क्रिमिनल इन्वेस्टिगेशन में हेल्पफुल रहता है तो इन दिस वे दिस पैलिनोलॉजिकल स्टडीज आर आल्सो हेल्पफुल इन द फॉरेंसिक science. So these importance basis of the forensic palynology that will pollens and spores productions and dispersions are important basis of the study of the forensic palynology. जो भी हम पोलन मोस्टली कहाँ पे प्रोड्यूस होते हैं उसका डिस्पर्शन कहाँ तक हो सकता है वो जो इन्वेस्टिगेशन है दैट विल बी हेल्पफुल in case of the forensic study. So pollen fingerprint of these different region that will be helpful to solving the different criminal cases. So forensic palynologist is trying to prepare the type and number of pollen grains in different regions and make their fingerprint so that will be helpful to solving or matching at the criminal sites and they will be helpful to find out the or analyze the different samples which are collected at the crime site and it will be helpful to find out the crime position or crime site. So knowledge of the pollen dispersals and productivity often plays a major role in solving the various legal problems. So palynological study ultimately helpful to solving the different criminal cases. These are some points in that when the pollen grains are having the single layer of the cellulose, at that time we have easily identified that pollen grains they are obtained from the plant that are grows in the water polluted region. जहाँ पे यदि हमें सिंगल लेयर सेल्यूलोज यदि पोलन ग्रेन्स में मिलता है तो वी कैन इजीली आइडेंटिफाई दैट पोलन ग्रेन्स आर द collected from the water polluted site that is aqua gamous regions. Then when the pollen grains are having the outer layer of the sporopollenin with the thick exine, at that time there will be confirmed that the pollen grains from the autogamous plant region. In this way, if the pollen grains are having the thick exine with the or some ornamentations like our exines having the excretions, at that time these pollens will be zoophilous plant region. Zoophilous, that means the pollen grains that will be dispersed with the help of animals. Then if pollen grains are having the thick exines with number of ornamentations or they are having no excretions, at that time these pollens from the anemophilous plant regions. So in this way, these are some tricks with this we can easily identify that the pollens that will come from the particular regions. And then in case of the solving the cases there will be collections of the hairs or cloth. जहाँ पे crime होता है तो वहाँ पे कुछ hairs या जो कपड़े वहाँ पे रहते हैं वो भी हम analysis कर सकते हैं. So with these woven cloths like the shirts, suits, ties, sweaters, fur या तो car seat, then car covers, also the exhaust straps. They can having some pollen or spores of that particular area. So when we collect this material, we can easily study out the pollens or spores which are present on that material, and we can help this investigation for the finding out the criminals. Then here gives some examples. So we just take first example. In that, a woman was killed in May during the trip in central Sweden. The body was with only shorts. The palynologist was asked to examine the dirt attached to the victim woman's shorts. So, studies of this pollen will be takes place on these shorts, which are where these ladies. So, studies of that pollens in this dirt sample suggested that she has been killed where the dirt is not contained. Pollens from these plants common in this area where the body was found. तो वहाँ पे जो भी जहाँ पे उस लेडीज की जहाँ पे बॉडी मिली थी वहाँ पे मोस्टली बहुत सारे पोलन्स प्रेजेंट रहे बट जब वहाँ पे उसके जो कपड़े थे कपड़े जब एनालिसिस किए तो वहाँ पे खाली उनको डर्ट मिली उसमें पोलन्स नहीं थे तो ये जो इन्वेस्टिगेशन है पैलिनोलॉजिकल यदि इन्वेस्टिगेशन हम करते हैं तो जो जो विक्टिम्स है जो क्राइम हुआ है क्राइम एनालिसिस करने में हमें हेल्पफुल रहता है तो अकॉर्डिंग टू दी डिफरेंट यहाँ पे एग्जांपल्स दिए हैं कि विद दिस एग्जांपल वी कैन इजीली फाइंड आउट और दिस इन्वेस्टिगेशन दैट विल बी हेल्पफुल टू फाइंड आउट सच अ crimes but there are some limitations in this study of the palynology, and there is a for study of the forensic palynology, there will be requirement of the some specialist. So without a specialist person, this pollen analysis study cannot be easy. So collections of the sample that will be also important. If the collections of these at the victim site correct collections of the material it is very important then facilities all the facilities for analyzing and that type of samples it is very important without facilities or without instruments we cannot do this study then these findings or the forensic pollens in the laboratories must be underground to maintain the forensic work and proper finding must be done. So if we can do the proper यदि हमें proper वो identification करना है तो वहाँ पे जो भी requirement है laboratory जो भी वहाँ का जो प्रिलिमिनरी डाटा है वो सब वहाँ पे चाहिए तब भी हम वो स्टडी कर सकते हैं और जो हमने स्टडी किया है वो जो भी क्रिमिनल केसेस है वो सॉल्व करने में हेल्प करेगा सो इन दिस वे दिस पैलिनोलॉजिकल स्टडी दैट विल बी आल्सो हेल्पफुल टू फाइंड आउट द or they will be helpful to find out the criminals in the forensic study. So in this way, these palynological study that will use or they are having the different applications and they are used in different fields. So in this way, your this palynological unit that will be over. You take the